Hello students, today we will discuss about BPT 23. Okay, so BPT 23 we will discuss for biology. Starting with the first question 101. It is saying that the science which deals with techniques of using live organisms or enzymes from organisms to produce products and processes useful to human is genetics, biotech, bioinformatics, or none of the above. So it is simple, you know that is simply biotechnology. That is what we are studying for. Now the next question is, a restriction endonuclease which always casts DNA molecule at a particular point by recognizing a specific sequence of 6 base pair. Consider this, okay? 6 base pair, that is hint second, okay? First option will be your correct answer for this question. You can check this also. Hint second. It is given in your NCERT book. Then the next question, it is saying that the specific DNA sequence in a chromosome which is responsible for the initiation of replication, okay? Cloning region, termination region, initiation region of origin of replication. Don't get confused with initiation region. That is nothing called initiation region. That is origin of replication. So fourth option will be your correct answer for this question. Then moving to the next question, that is Taq polymerase is used in polymerase chain reaction. Why? Because it becomes inactive at high temperature. Then there is no use because it becomes activated. It remains activated in high temperature. So it is not the answer. It makes other enzyme active at a high temperature. No, this is also not the reason. Then what is actually the reason? It remains active during the high temperature. This is the absolute reason. It obtained from a thermostable virus. This is actually true, but not the, this is not for it is used. So third option will be your correct answer for this question. You can also read this from here. Taq polymerase active during high temperature. Then the next question 105. It is saying that 105, right? It is saying that which of the following enzyme is known as genetic glue? Genetic glue, which one is? DNA polymerase? No. Alkaline phosphatase? No. These are a different sort of enzyme. But DNA ligase, which joins the DNA. Okay, that is means the genetic glue. Glue means, okay, you know. So third option, genetic glue is DNA ligase. Not all of the above. Next, moving to the next question. That is question number 106. A small chemically synthesized oligonucleotides that are complementary to the regions of DNA at the three end used in PCR are primer, dimer, small strands or large fragments. So these are basically primer. First option will be your correct answer. The primers are the small chemically synthesized oligonucleotide that are complementary to the regions of DNA. Then question number 107. Bombardment of high velocity microparticles of gold or tungsten coated with DNA on target cells are biolistics, microinjection, electroporation or bombing. This question is very important. This can come in NEET 2025, I think. That there are different ways to introduce alien DNA into host cell, we know. So this process is basically called biolistics. So first option will be your correct answer for this question. Moving to the next question, that is question number 108. Let's see what it is saying. The most common matrix is agarose gel, and natural polymer is used in gel electrophoresis is extracted from animal, fungus, seaweeds, or what is none of this. So this is extracted from seaweeds. We know that. So third option will be your correct answer. Then moving to the next question, question number 109. Let's see what it says. It is saying that a typical agarose gel electrophoresis showing migration of a set of DNA fragments. So you can see this is the migration of different DNA fragments. So which of the following statement is correct regarding this? Only lane first shows undigested DNA fragment. Okay, lane shows, you can see there is no movement, so that means the DNA remains undigested. So this is the correct option actually. And only the lane 4 shows digested DNA. No, these are all digested DNA. This is not the correct answer. Lane 1 to 3 shows, lane 2, 3, 4 all shows digested DNA. This is also not correct. Only lane 4, this is not correct, right? So first option will be your correct answer for this question. By you can see, you can deduce from the picture. Then question number 110. It is saying that the figure below diagrammatic representation of E. coli vector PBR322. Which one of the following options correctly identifies its components? Okay, let's see. In third, eco-ROI selectable markers. So this is not the correct one. Ampicillin and tetracycline, you can see this These are antibody resistant gene. Absolutely correct. So second option will be your correct answer for this question. Let's see also other option, what other saying. ORI, original restriction or origin of replication, you know. ROP, this is also, it is a semantic question, is not the thing. Okay, this is not correct. Then 111. It is saying that the diagram below refers to recombinant DNA technology. Identify A to D. Okay, so A basically represents echo R1, B represents echo R1 here, B also represents echo R1. This is a good question, I will say. Okay, C represent DNA ligase, and D represent transformation. You can see this is transformation process. So our correct option will be second option for this question. Moving to the next question, that is saying the basic procedure involved in the synthesis of recombinant DNA molecules is depicted below. The mistake in the procedure. Let's see what it is saying. Okay, this is the procedure, right? So it is saying that enzyme polymerase is not included. We can see that polymerase restriction endonuclease, mammalian. So this is not correct. Then the mammalian DNA is shown double-stranded. Mammalian DNA is double-stranded, so nothing wrong in this. Only one fragment is inserted. So this is also not correct. Two different restriction enzymes are used. Restriction endonuclease 1, restriction endonuclease 2. Okay. This is all the mistake actually. Okay, so DNA technology, endonuclease 1, endonuclease 2, because you can't use 2, that will complicate the process. Okay, so fourth option will be your correct answer. Moving to the next question, this is question number 113. It is saying that which of the given statements is correct in the context of observing DNA separated by agarose gel electrophoresis. DNA can be seen under visible light. DNA can be seen without extending the visible light. Ethidium bromide extended DNA can be seen in visible light. Ethidium bromide extended DNA can be seen under exposure to UV light. This is the correct one. Okay. Fourth option will be our correct answer. This is given in NCERT. This is very easy. Then question number 140. The following statements describe the characteristic of enzyme restriction endonuclease. Identify the incorrect statement. So you have to find the incorrect statement. The enzyme cuts DNA molecule at the identified position within the DNA. Absolutely correct. Then the next. The enzyme binds DNA at specific sites and cuts only one of the two strands. No, they cut both the strands. This is not correct. The enzyme cuts sugar phosphate backbone. Correct. The enzyme recognizes a specific palindromic sequence. It is also correct. So our correct option will be second option, because this is the incorrect statement. Question number 150. It is saying that given below are the four statements about the separation of DNA fragments using gel electrophoresis. Identify the incorrect statement. Okay, so you have to find the incorrect statement. First is saying that DNA is a negatively charged molecule. Correct. So it is loaded in anode. No, this is loaded in cathode terminal. So this is not correct. And DNA fragments travel along the surface of the gel, which concentration does not affect the movement of DNA. Yeah, it is correct. Then the smaller the size of the DNA fragment, the larger the distance it travels through it. This is also correct. This is not correct actually. The DNA fragment, it does affect the movement. Okay, so this is not correct. Then pure DNA can be visualized directly. No, not directly. It should be ethidium bromide stain. So just we have seen this question. This is also not correct. So which are the incorrect? A, B, D are incorrect. Fourth option will be our correct answer. Moving to the next question, question number 116. It is saying that read the following statements and choose the incorrect one. We can introduce. Okay, so you have to read this question first. Incorrect one. Okay. So here incorrect one you need to find. We can introduce only one or a set of desirable genes without introducing undesirable genes into the target organism. Absolutely correct. Then sexual reproduction preserves, no, it doesn't preserve the genetic information, it changes. That's why the sexual reproduction creates variation. Then plasmids show the autonomous mode of replication. It is correct actually. And plasmids have only one copy. No, they have several copies. So which are incorrect ones? So the incorrect ones are B and D. Third option will be your correct answer. Then moving to the next question, that is 117. 170. It is saying that in cloning vector PBR322, BAM H1 site is located in. This is, you have to visualize the diagram. Okay, this question have came several times in NEET. It's a very good question. Amplicillin resistant gene, tetracycline resistant gene, kanamycin resistant gene, or chloramphenicol resistant gene. So this is basically located in tetracycline resistance gene. Second option is our correct answer. Then moving to the next question, question number 118. A restriction endonuclease breaks the bond between base pairs of the DNA molecule, base pairs of the DNA RNA, sugar phosphate components of nucleic acid. This is the bond they break. So third option will be our correct answer. Then question number 119. It is saying that when was the first recombinant DNA molecule constructed? 1962, 1972, 1982, 1992. So this first recombinant DNA molecule constructed in 1972. Second option. Moving towards the next question, question number 120. It is saying that select the correct options. Okay, for the statements. The source of the restriction enzyme hint 3 is hemophilus influenza. So this means hemophilus influenza. This is correct one. In biologistic method of gene transfer, microparticles made of gold or tungsten are coated with foreign DNA. Correct. Micro-injection method for injecting recombinant DNA is used for animal cells. Absolutely correct. Then primers are chemically synthesized oligonucleotides that are complemented to the regions of DNA in PCR, that is polymerase chain reaction. This is also correct. So how many statements are correct? All are correct. So first option will be your correct answer. Moving towards the next question, question number 121. Let's see. In recombinant DNA technology or genetic engineering, elution means. So what does elution mean? So elution basically means that separated bands of DNA cut from the gel and extracted from the gel pieces. That's the same thing. Okay. So this is the separated bands of DNA are cut out from the gel and extracted from the gel piece. Second option will be the correct answer. Then question number 122. Which of the following tools of recombinant DNA technology is incorrectly paired with use? Okay, let's see. Restriction enzyme, production of restriction nucleotide fragment. Okay, so this is correct. And DNA ligase joins the DNA, that doesn't cut the DNA. Okay, DNA polymerase used in polymerase chain reaction to amplify sections of DNA. Correct. Then reverse transcriptase, production of cDNA, complementary DNA from mRNA. Correct. That's why the name is reverse transcriptase. Second option is our incorrect one. Then question number 123. If a recombinant DNA bearing gene for amplicillin resistant is transferred into E. coli cell and the host cell are spread on the agar plates containing amplicillin, then what will happen? You can just apply the logic. Okay. What will happen? Recombinant DNA, just look at this. If a recombinant DNA bearing gene for amplicillin resistant is transferred into E. coli cell and the host cell is spread on the agar plates containing ampicillin, then transformed recipient cell will grow and untransformed recipient cell will definitely die. So our correct answer will be third option. Question number 124. Which vector is commonly used in transfer of genes in crop plant? Plasmids of Bacillus subtilis, bacteriophages, TI plasmid of Agrobacterium, E. coli feces. TI plasmid of Agrobacterium is most preferably used. So third option. Then question number 125. Restriction endonucleases are used in genetic engineering because they can degrade harmful proteins, they can join DNA fragments, they can cut DNA at variable sites, they can cut DNA at a specific ways where you want to. So that's why it is used. So fourth option will be your correct answer. Now moving to the next question, that is restriction assertion design. Okay, so restriction enzyme cuts the strand of DNA to produce sticky ends, and the reason is saying that stickiness of the ends facilitates the action of enzyme DNA polymerase. But this reason is false. Okay, This reason is false. Assertion is true. Assertion is true. Restickiness of the DNA ends facilitates the action of the DNA ligase, not DNA polymerase. Okay. So third option will be our correct answer. Now moving towards the next question, that is question number 127. Gene amplification can be done by polymerase chain reaction, obviously. Then in PCR, multiple copies of the gene of interest are synthesized. This is absolutely correct. Now if we put a because there, now we read the whole statement. Gene amplification can be done by PCR because in PCR, multiple copies of gene of interest are synthesized. So both assertion and reason are true, and reason is the correct explanation of assertion. So first option will be our correct answer. Then question number 128. Oh my god, there are so many assertion of reasons questions there. Okay. So assertion, restriction endonuclease are called molecular scissors, absolutely correct. Then reason is given when fragments generated by restriction endonuclease are mixed, they join together due to sticky ends. Okay, this is also correct, but the reason is not actually describing why they are called molecular scissors. So that's why both are correct, but reason is not the correct explanation of assertion. Second option will be your correct answer. Then moving towards the next question, question number 129. It is saying that assertion, the process including separating and purification of the product collectively refers to as downstream processing, correct? Then the downstream processing is the same for all product. No, this is not same for all product. So third option will be your correct answer for this question. Then the next question, assertion reason 130. It is saying that recombinant DNA technology requires both nuclease and ligase, absolutely correct. Then what is the reason the same? Reason is saying that nucleus is used to join. No, nucleus doesn't join the DNA strand. It is the work or function of ligase. So third option will be your correct answer. Then question number 131. This is the most easy assertion reason, I think. You don't need to think much. In recombinant DNA technology, human genetics are often transferred into bacteria or yeast, correct? Reason is given, both bacteria and yeast multiply very fast to form huge population that express the desired gene, correct? If we put a because here and now read the whole statement, we will see that the reason is correctly describing the assertion. So first option will be your correct answer. Then question number 132. What it is saying that 132. Alternative selectable markers were developed to differentiate non-recombinants from recombinants based on ability to separate them according to size, no. Ability to produce color in the presence of chromogenic substances. That's why they are being differentiated. So second option will be your correct answer. Then question number 133. If DNA is inserted within the coding sequence of beta-galactosidase enzyme, then what will happen? Non-recombinants will give blue colored colony in presence of chromogenic substrate. If DNA ligase is coding beta-galactosidase, okay, so this option I think is correct. Absolutely, this is correct. So first option will be our correct answer. Then moving towards the next question, question number 134. It is saying that Taq polymerase is obtained from Bacillus thuringiensis, Thermus aquaticus, Salmonella typhimurium, Escherichia coli. So it is obtained from Thermus aquaticus. Second option will be our correct answer. Then question number 135. What does it says? It is saying that EcoR1 recognizes the palindromic sequences. Which is the palindromic sequence of Gatach? I remember like this, Gatach, G-A-T-T-C. Okay. So this second option will be our correct answer. In my time also I remember like this. So you can also remember like this. In question number 136, I guess. Okay, 136. Sorry. So what is downstream processing? Process of separation of DNA fragments, no. Process of joining the vector and the dose DNA, no. Process including separation and purification of the product is the absolute answer. Third option. Then 137. The construction of the first recombinant DNA was done by, I think this can be like given by you. This can be given as a scientist name. So this is a extra mark question. So done by Stanley Cohen and Herbert Boyer. So first option will be correct answer. This can come in match the following for NEET 2025, I think. Then question number 138. To make cell competent to take up DNA, heat shock is given to cells. The temperature of shock is 30 degree centigrade, 42 degree centigrade, 60 degree centigrade or 90 degree centigrade. So this shock temperature is given, that is like to make the cell competent. Heat shock is given at 42 degree centigrade. So second option will be the correct answer. In question number 139. If the bacteria if the bacterium does not have any insert, then the presence of chromogenic substance give red color colonies, colorless colony, blue color colony or green colony. So if the presence of chromogenic substances gives blue color colony if the plasmids in the bacteria does not have an insert. So what will answer? Blue colonies, third option. Then moving towards the next question. That is 140. Based on the nomenclature of the molecular seizure, match the following columns, choose the correct option. This can come in NEET 2025. NEET 2025. Okay. So what is co? Co is the species. Then what is the eco, that is E. So what is the E? E is basically the genus. Then eco, R. R is basically what it is, the strain. And first is the order of extraction. So correct answer will be option number first. Then moving towards the next question, question number 141. Match the column with column first with column second. ELISA, you know that, antigen antibody reaction. ELISA, nothing else. Enzyme linked immunosorbent assay. PCR polymerase chain reaction, that is used for what? Gene amplification. Ballistics, okay. Ballistics is what? Gold coated with DNA. Gold coated with DNA. And microinjection reaction, what does it? Direct introduction of recombinants. So our correct answer will be option number second. Then question number 142. Which of the following tools of recombinant DNA technology is incorrectly paired with its use? Okay, let's see. First option is given, restriction enzyme, production of RFLPs, correct. And DNA ligase that cuts the DNA, that doesn't cut the DNA, it's just joins. DNA polymerase, correct. Reverse transcriptase is also correct. Second option. Now we will move towards the question number 143. This is a statement based question. We will read the statement and try to find out our answer. Okay. It is saying that statement first, in gel electrophoresis, pore size is related to the concentration of the gel. Absolutely correct. Then the concentration of gel is inversely proportional to the pore size. It is also correct. So both statements are true. So first option will be our correct answer. Moving to the next question, question number 144. This is also statement based. PCR primers must have a self-complementary region. Must not have, like this is not correct. It should not be must. They can have or not have. Then complementary base pairing will form hairpin structures that are suitable as a primer. So if a PCR primer has a self-complementary region, then it will form hairpin like a structure, but they are not suitable as primer. So this is also not correct. So both statements are incorrect. Second option is our correct answer. Then question number 145. It is saying that separated DNA fragments in gel electrophoresis are exposed to UV radiation, followed by staining. First you have to stain, then you have to expose. So this is wrong. Then pure DNA fragment will be visible by themselves. You need to stain to see them. So both are incorrect. Second option is our correct answer. Then question number 146. Statement first is saying that unless one cuts the vector and the source DNA with different restriction enzyme, a recombinant vector molecule cannot be created. It can be created. Okay, so we can see this is not correct. And when cut by the same restriction enzyme, the resultant DNA fragments produce sticky end and this cannot be joined together. This can be joined together. Okay, this is not correct. So both statements are incorrect. Second option is our correct answer. And question number 147. How many statements are incorrect? It is saying that DNA cannot pass through the cell membrane directly, correct? DNA can pass through the cell membrane directly. So statements are incorrect. Okay. So it cannot pass through all membrane. So DNA can pass through all membrane directly. So this is not correct. Okay. Because DNA is a hydrophilic molecule. Then DNA is hydrophilic in nature, correct? DNA is both hydrophilic and hydrophobic in nature. So this is also not correct. So second option is our correct answer. Then question number 148. 140, let's see what it says. And select the correct option. How many statements are correct? Okay. A thermostable DNA polymerase is used as PCR for repeated amplification. Correct. It remains active. It is also correct. Then the polymerization stage of PCR is when Taq polymerase, which was isolated from thermophilic bacteria Thermus aquatica, synthesis the region of DNA between primers. Okay, this is also correct. When double-stranded DNA is denatured by high temperature, the Taq polymerase becomes inactive. No, this remains active. That's why it is being used. So 3 statements are correct. Third option will be your correct answer. Question number 149. Which of the following statements are correct? 149. Let's see. If the following statements are correct, let's see the question now. Yeah. So bacteria are treated with a specific concentration of divalent cation such as barium for transformation. So this barium is not commonly used for this purpose basically. Okay, so cation, divalent cation like calcium can be used. Then this is done to take up the DNA by bacterium since DNA is a hydrophilic molecule and cannot pass through the cell membrane. Correct. Then bacteria are treated with the specific concentration of not trivalent, obviously not, so this is incorrect. And another technique for injecting DNA straight into animal's nucleus is known as microinjection. Correct. So the correct statement B and D, third option will be your correct answer. So now we will moving towards the last question of this botany part, question number 150. It is saying that which of the following statement is correct? For the multiplication of any alien piece of DNA in an organism, it needs to be a part of chromosome that has a specific sequence. Okay, so let me see the. This one is the correct one actually. Then in a chromosome, this specific sequence is known as origin of replication. It is also correct. Then in a chromosome, this specific sequence is known as the origin of replication, which is responsible for translation. No, not correct. This is the origin of replication is for copy number, making copies. For the multiplication of any alien piece of DNA in an organism, it needs to be a part of a chromosome that is a non-specific sequence. That need to be have a specific sequence. So the correct option first, A and B. Now we are moving towards the zoology portion, discussion for zoology. Question number 151. The prerequisite for biotechnological production of antibiotics is to search for antibiotic producing microorganism, absolutely correct. To isolate the antibiotic gene, correct. Then to join antibiotic gene with E. coli plasmid, it is also correct. So all of the above are correct answer. Fourth option will be our correct answer. Then question number 152. It is saying that the process of RNA interference developed a transgenic tobacco plant which was resistant to infection of algae, skin desmas, fungi, fusarium, bacteria, bacillus thuringiensis, nematode, melodiguin incognita. Okay, so this I think out of NCERT, so I will not no, I think this is given in NCERT. I think this is given in NCERT. If this is given in NCERT, then you have to study. I forgot that. Oh, yeah, this is given in NCERT. The fourth option will be our correct answer. Melodiguin incognita. Nematode is given actually. By a nematode. Not the name is maybe not given in new NCERTs. Question number 153. It is saying that Bt toxin kills the insect by blocking the nerve conduction, damaging the surface of trachea by creating pores in the tracheal system, or by creating pores in the midgut. So, creating pores in the midgut. Fourth option will be our correct answer. Then question number 154. It is saying that correctly match column first with column second. Bt cotton 155, right? So, Bt cotton is matched with cotton bollworms, nematode resistant tobacco, RNA interference technique, super mouse growth hormone gene, rosy, you know that human alpha lactalbumin. So, our correct option for this question will be option number first. Then question number 156. Match the column, same. So, we need to see. Most of the, from this chapter, you will get match the column type of question mostly. That's why so many match the column are questions given in your unit test. Biopiracy, what does it mean? It means illegal exploitation of biological material. Okay. And biopatent right granted to the biological entities. Gene therapy is efforts to correct the non-functional gene. And RNAi, that means gene silencing. So, our correct answer for this question will be option number second. Now, moving towards the next question, question number 156. Let's see what it is saying. So, cry 1, this is very confusing. I am choosing this as the NEET 2025 paper. This can come in NEET 2025 paper. So, cry 1 AC is cotton bollworms, cry 1 AC. Cry 1 AB is cotton corn borers, TI plasmid agrobacteria. Only these are given. Yeah, actually, like this. Okay. So, our correct answer for this question will be option number 4th. Then question number 158. Bt toxin. You know that Bt toxin is crygene. RNA interference is silencing of mRNA. RDT, recombinant DNA technology. DNA RNA probe. Gene therapy, complementary DNA. Adenosine deaminase, complementary DNA. Gene therapy, you know that first happen in a gull. So, this question option number 4th. Then moving towards the next question, that is question number 160. What it is saying? Transgenic plants are the ones grow in artificial medium after hybridization in the field. No. Produced by a somatic embryo in an artificial medium. No. Generated by introducing foreign DNA in a cell and regenerating a plant from that cell. Absolutely correct. That's why it is saying that transgenic plants, genes are being transferred and produced after protoplast fusion in an artificial node. So, third option will be our correct answer. Next question number 161. Which of the following statements is correct about Bt toxin? Let's see what are the options given. So, the concerned bacillus has antitoxin. Not correct. Then the inactive protoxin gets converted into active form in the insect gut. It is also correct. This is correct actually. Then let's see other options. Bt protein exists as an active toxin. No, it does not exist as an active toxin in the bacillus. It gets converted. Then if the activated toxin enters the ovaries of the no, this is not correct. It's just activated in the gut. Second option is our correct answer. Then question number 162. 162. What does it say? Read the following statements and which of the two statements has mistake. So, we need to find the incorrect statement. Let's see. Now, the first transgenic buffalo, Rosie, produced milk that was human alpha lactalbumin enriched. So, the milk contained So this is not correct. Then restriction enzymes are used in the isolation of DNA from other macromolecules. This is not correct. Then downstream processing is one of the steps of recombinant DNA technology. Correct. DSAM pathogen vector are also used in transfer of recombinant DNA technology into the host. So these are also correct. So which of the following are incorrect? A and B. So first option will be our correct answer. Then moving towards the next question, that is question number 163, I guess. Yeah, 163. Which of the following correct statements regarding golden rice? It is a vitamin A enriched with gene from daffodil. Absolutely, this is the correct one. So you can just take this option. First option will be your correct answer. Then question number 164. Which of the following is the correct statement? In India, GEAC will make decisions regarding the validity of GM research and safety introducing GM organisms for public service. Correct. Golden rice is distinct for its unique aroma and flavor, and 27 varieties of golden rice are grown in India. This is not correct. In India, basmati rice, actually 27 varieties of basmati rice, I guess, are grown in India. The Indian Parliament did not amend, it does amend in 1990. This is also not correct. So first option is our correct answer. And just read the statements. I am not wasting any time. Question number 165. Select the true statements regarding insulin. This can also come in NEET 2025. Select the true statements from the following regarding insulin. Insulin from animal sources may develop allergies in some patients. Correct. Recombinant insulin does not introduce unwanted immunological response. Correct. C-peptide is not present in mature insulin. Correct. Insulin can be administered orally to the diabetic patient. No, you know that insulin injection is given. So this is not correct. So A, B, C are correct only. Third option will be your correct answer for this question. Then question number 166. Read the following statements and how many statements are incorrect for applications of GM crops. Okay. The first statement is given and that made crops more tolerate to biotic states such as cold, drought, salt and heat. Correct. Then one second, this is saying that these are biotic states. So these are not biotic states, right? These are abiotic states. So this option will not, like, this is the incorrect statement. Okay, so this is incorrect statement. I'm sorry for this, that I have not seen the biotic. These questions like simple word you can see. Reduce reliance on chemical pesticide. Correct. Help to reduce post-harvest loss. Correct. Increased efficiency of mineral uses by plants. Correct. That's why this the GM crops is implemented or that is being developed. So how many are incorrect? One is incorrect only. So first option will be our correct answer. Then question number 167. 167. Where it is? Yeah, got it now. Statement first. A gene from Bacillus thuringiensis is incorporated into the plant genome to increase their yield. This is incorrect. And Bacillus thuringiensis has a Bt toxin producing gene which kills insect larvae. It is absolutely correct. So fourth option will be our correct answer for this question. Then question number 168. 168. Statement first. Human insulin can be produced by protozoa. Human insulin can be produced by protozoa. No, I don't think so. This is incorrect. Okay, so insulin from different animals caused allergic reaction in human. Absolutely correct. So fourth option will be our correct answer for this question. Then question number 169. 169. Statement first. A functional adenosine deaminidase, a complementary DNA is introduced into the lymphocytes of patients suffering from ATA deficiency. This statement is correct. Then The patient does not require periodic infusion of such genetically engineered lymphocyte. They does need periodic infusion of genetically engineered lymphocytes. So third option will be our correct answer. Then question number 170. It is saying that a statement first, ELISA is based on the principle of antigen-antibody interaction, correct. Then infection by statement second, infection by pathogens can be detected by detecting the antibodies synthesized against the pathogen, correct. So both are correct, first option will be our correct answer. Then question number 171. It is saying that the milk of transgenic cow Rosie is not better than the natural cow milk. It is actually better. So this statement is not correct. Okay. Then statement second, it produces human alpha lactalbumin enriched. This is correct. The first is incorrect, second is correct, fourth option. Next question number 172. It is saying statement first, Bacillus thuringiensis is not killed by its toxin. Yes, it is correct. Then Bt toxin protein exists as inactive protoxin. It gets converted in. So first option will be a correct answer as both the statements are correct. Then question number 173. What it is saying that the genetic defect adenosine deaminase deficiency may be cured permanently by enzyme replacement theory, periodic infusion of genetically engineered lymphocytes having functional ADA complementary DNA, administering adenosine deaminase activators. These are all correct, but these are not the permanent cure. These are temporary cure. What is the permanent cure? That is introducing bone marrow cells producing ADA into cells at early embryonic stage. So this fourth option will be our correct answer. Then question number 174. An improved variety of transgenic basmati rice is completely resistant to all insect pest and disease of paddy. I don't know, not correct. Gives high yield but has no characteristic aroma. Why its name is basmati rice? Because its aroma. If you eat some biryani of basmati rice, then you will get to know what is basmati rice, the long grains, okay. Then does not require chemical fertilizers and growth hormone, incorrect. So it gives high yield and rich in vitamin A. So fourth option is our correct answer. Then question number 175. It is saying that genetic engineering has been successfully used to produce transgenic cow Rosie, which produces high fat milk for making ghee. No, this is called alpha lactalbumin. An animal like bulls for farm walk as they have a superpower. I don't know what kind of options they have given. Not correct. And transgenic mice for testing safety of polio vaccine before using human. Correct. Transgenic models for studying new treatments for certain cardiac disease. You can't study like this. So third option will be the correct answer. And question number 176. The process of RNA interference has been used in the development of plant resistant to nematode, fungi, virus or insects. This is nematode, resistant to nematode. First option. So I have previously this question has been asked, but there the name is given. Don't get confused with the name. So just remember it is a nematode. Then question number 177. Bacillus thuringiensis forms protein crystal that contain insecticidal protein. This protein does not kill the carrier bacterium which is itself resistant to toxin. Not correct. Okay. Then binds with epithelial cell of the midgut of the insect pest, ultimately killing it. Correct. Then is coded by several genes including the gene cry. Not correct. Is activated with alkaline pH. Then second option is our correct answer. Question number 178. What it is saying? That transgenic animals are those which have foreign RNA its all cell, foreign DNA in some of itself, foreign DNA, obviously. Third option. Very easy. Biopatents are. What are biopatents? Right to use invention, correct. Right to use products, correct. Right to use biological entities, correct. Right to use the process, correct. So fourth option is our correct answer. Then question number 180. Memory based question, simple. If you don't remember, then just skip. But you can't skip. In 2021 I appeared, I got 360 out of 360. It was a huge achievement for biology. I mean, oh my god. But now, like, I have seen like 14k people or more than that get 360 out of 360. So you need to score 360 out of 360 in biology, or 355 is also okay. Below 350, if your bio score is like this, below 350, then you can't secure government medical college, good government medical college in these competition times, because in physics or chemistry, you know that the one silly mistake can cause like a huge hemorrhage. Like if you got silly mistake, then you got, suppose I'm saying, you get 20 marks less in physics and 20 marks less in chemistry. And if your biology marks is 350, then you will get just 10 marks. Then still your score stands 670. I think 670. 670 is a good score. You will get a very good college. But below than that, below 630 or 640, what kind of college you will get in these times? Like if you saw the pattern of NEET 2024, the scores in 640 is a very good marks. Even 610, 600 is a very also good mark. But this does not apply in this time. So you need to work hard. You need to score more. You need to try to improve your mistakes. That's why we are giving so many tests now. That is the reason we are giving so many tests. Why there is the reason to attend 36 of tests and more than that. This is the reason, that so that you can get so much of practice and ultimately improve your score. 360 out of 360 is that what I want. Okay. So let's stick to the question now. At present, about how many recombinant therapeutics have been improved for human the world for use? Okay. So that has been like 30 recombinants. Third option. In India, all are not like, not all, everything is used. Then question number 181. A whole plant could be regenerated from explant. This technique is known as plant tissue culture. Third option. Don't get tempted by this option, totipotency. Okay, that is different. Then question number 182. 182 is saying that two stages of insulin, stage P and stage Q is given. Okay, this is mature, this is immature. You can see that C peptide. So select the correct statement. Stage P is the earlier active stage of insulin. And then stage Q is the active and earlier stage of insulin. Stage Q is referred to as proinsulin. Stage P is the proinsulin. Stage P is an inactive stage. So fourth option is our correct answer. Okay, inactive insulin, proinsulin. Then question number 183. Assertion reason. Let's see what it is saying. 183. Scientists have isolated single cell from plant. Okay, this is correct. Then reason, isolated protoplast from two different varieties of plants can be fused to get hybrid protoplast, but they cannot be further grown to form a new plant. They can be grown. So here assertion is correct, but reason is not correct. So third option is our correct answer. Then question number 184. The traditional breeding technique succeed in keeping pace with demand and providing sufficiently fast and efficient system for crop improvement. I think if anyone can answer this question, this is not correct. Like traditional, nothing is like, except some of the things, everything is being replaced by artificial intelligence or some robots or some improved technology. Okay, the capacity to generate a whole plant from a cell explant is called totipotency. This is plant tissue culture. So our option, fourth option will be our correct answer. Then question number 185. 185. Let's see what our question says. We are on the verge of end. Assertion: a genetic modification of organisms can lead to unexpected outcomes when they are introduced into ecosystem. Correct. Ecosystems are complex and new traits from GMOs can interact in unexpected and unfavorable ways. This is also correct. So now if you put a because here, now read the whole statements, you will get that assertion reason both are true and reason is the correct explanation of assertion. So first option is our correct answer. Then question number 186. 186. A probe, which is a molecule used to locate homologous sequences in a mixture of DNA or RNA molecules, could be a single-stranded RNA, a single-stranded DNA, either DNA or RNA, can be single circular ssDNA, single-stranded DNA, but not single-stranded RNA. So this is like either DNA or RNA. Third option is our correct answer. And the site of production of adenosine deaminodase in our body, let me guess. Sorry, let me think about this question. This is the easiest question. This is the obviously lymphocyte. What you are infusing? Lymphocyte. So in RNA interference, the silencing of a specific mRNA due to a complementary double-stranded RNA molecule that binds to and prevents the mRNA translation. That is the whole procedure of RNAi, that is the RNA interference. The silencing of a specific mRNA due to double-stranded DNA, no, double-stranded RNA. The silencing of a specific mRNA due to tRNA. No, tRNA is the adapter molecule, you know. So, first option is our correct answer. Then question number 189. 189. The two polypeptide of human insulin are linked together by hydrogen bond, phosphodiester bond, covalent bond, disulfide bridges. Tell me, what is this answer? Disulfide bridge, fourth option, right? So, fourth option is our correct answer. Then question number 190. The statement first. The proteins encoded by the Cry1SC and Cry1AB, cotton borer and Cry1AB controls cotton bollworms. I don't think so. This is just the reverse thing. Okay. You can see this. The protein encoded by the gene Cry1SC and Cry2AB control has cotton bollworms, and Cry1AB has the corn borer. So, Bt toxin are plant group of specific. So, Bt toxins are not specific to plant groups. They are specific to insect pests. So, both are incorrect. Second option is our correct answer. Question number 191. Identify the correct statement with respect to the development of transgenic animals for their study of the disease. Transgenic animals are designed to increase our understanding of the role of the genes in disease, correct? That's why they are being developed. Animals are developed as models for human diseases to investigate new methods of treatment, because you can't apply on humans, that is prohibited. And models exist for Alzheimer and cystic fibrosis, correct? So, fourth option is our correct answer. It is a little out of NCERT, but you can answer this if you have some common knowledge. Then question number 192. Assertion. We can produce transgenic bacteria to yield the desired product, correct? Human insulin can be produced by bacteria, correct? But this reason is not explaining the assertion that why we can use, how we can use, whatever like that, nothing. This is just off topic. So, second option is our correct answer. Then question number 193. It is saying that Flavor Savor tomato was the first transgenic commercial crop to enter into the market, correct? A roundup variety of soybean was prepared through breeding, no. So, third option is our correct answer. Simple. If you get both correct, then it gets confusing. If you get one of them incorrect, then obviously it could be either 3 or 4. Then question number 194. Eli Lilly is an American company, prepared 4 DNA sequences. No, they doesn't prepare 4 DNA sequences. They just prepare 2 DNA sequences. So, this is already becoming incorrect. I don't need to read the full statement to waste my time. And reason, but you should obviously read the statement, because you don't know, there are some twist after that. Chains A and B are produced separately, extracted and combined by creating disulfide bonds to form human insulin. Not correct. So, both assertion is not false. Both option is our correct answer. Then question number 195. It is saying that the first clinical gene therapy was given in 1990 to a 4-year-old girl with adenosine deaminodase deficiency, correct? The enzyme adenosine deaminodase inhibits the immune system of the human beings. It does not inhibit. So, reason is false. Third option. It just helps. In question number 196. What it is saying? The following figure shows the process of maturation of proinsulin into insulin. Identify the product A. So the identify product A. A is basically A. This is on is there, right? So this is the free C peptide. Okay, so fourth option is our correct answer. Then question number 197. Genetic modification has made crops less tolerant to abiotic stresses. It becomes like more tolerant. Not correct. Reduce reliance on chemical pesticide. Correct. Help to reduce post-harvest losses. Correct. Both options are fourth option. I think this kind of questions we already solved. Limit of questions. Like we are not getting enough questions to put. Like good questions. Obviously, you need to solve the best questions possible. In question number 198. Most of the industrialized nations are poor in traditional knowledge as well as financial. I don't think so. Yeah. This is not correct. Okay. They are rich in traditional knowledge but poor financially. Not correct. Rich financially but poor in biodiversity. This is the absolute thing. Suppose we are like developed country, Germany, America, like this. Okay. Poor financially but renowned. So third option. Then question number 199. Cultivation of BT cotton has been much in the news. The prefix BT means barium treated cotton seeds. No. Bigger thread variety of cotton with better tensile strength. Oh my god, what is that? Not correct. And then produced by biotechnology. No. Carrying an endotoxin gene from Bacillus thuringiensis. That's why B. Fourth option. Fourth option is our correct answer. Then question number 200. Okay. 200. Last question. The use of bioresources by multinational company and other organizations without proper authorization from the countries and people concerned without compensatory payment is known as biopiracy. The second option is our correct answer. So this concludes the whole explanation of BPT 23. I hope you understand it. Thank you.